Okay, everyone, this is chapter 16. Chapter 16 is referring to immunopathology or immune diseases. So whether your immune system is overreacting inappropriately or underreacting inappropriately, the term for that, the correct term would be immunopathology. Okay. First, we're going to talk about the hypersensitivities, and then we have later on the immunodeficiencies or hyposensitivities. So hyper, too much, right? Type 1 hypersensitivities. These are going to involve IgE. This could be causing atopic reactions or atopic reaction, however you want to say that. But that is going to be a local reaction. That would be an example, would be typical allergies and asthma. Those are local, right? Anaphylactic shock is systemic. So those are the differences there. We have atopy and anaphylactic shock. Both of those are driven by IgE responses. Both of these have to have a sensitizing reaction, just like we just talked about in chapter 15. You have to have seen the thing before and have memory of it in order for you to react so quickly to it. All right, types of allergens, which are things that can cause these responses, could be inhalants like pet dander, contactants like latex allergies, or lotions or detergents, injectants, kind of what it sounds like, but that also includes bee venom. Bee venom is an injectant because it gets injected into you, but naturally. And then ingestants, that would be foods like shellfish, peanuts. Sorry, you can tell I don't have any food allergies. I do, but it's like a weird one, so we don't need to talk about that. All right. And then for treating these, if we're talking about our local infections, the best treatments are going to be our antihistamines or our corticosteroids. These are going to lower that immune response, basically tamping it down. The problem with some antihistamines like Benadryl, which is diphenhydramine, they can cross the blood-brain barrier, which would be great if we're trying to treat something in the brain. But the problem is that because it does that, it has that sort of sleepiness that you can get as a result. So, right. And then if you're trying to treat the systemic version, anaphylactic shock, you're going to treat that with an EpiPen, right? So that's epinephrine or adrenaline, same thing. Type 2 responses. Type 2 hypersensitivities involve IgG and or IgM. These will cause complement-mediated lysis. We talked about complement, right? Classical complement pathway. We have antibodies that will be binding onto whatever is going on. And then we'll trigger the complement pathway. Complement proteins that are just flowing around in the bloodstream start, splicing and dicing. That's the best way to look at it, I guess. But like some of them come together and become reactive. Some of them split apart to, make the next thing reactive and so on and so forth. But it's a cascade that essentially ultimately leads to inflammation, opsonization, which is helping phagocytosis, right? And complement-mediated lysis, cytolysis. So it's poking holes in cell membranes to release their content. In this case, we're treating the bad guy is the blood group that would, be incompatible with ours. We talked about ABO blood groups. If you are an A, if you are an A, type A blood type, you have antibodies. Geez, I wish I could write better with this. You are antibodies against B. If you are a B, you have anti-A. If you are an O, you have, obviously, if you're an A, you're not going to have anti-A because that's self to you, right? And a B, same thing. You're not going to have anti-B because that's self to you. O, though, O doesn't have any specific marker on there. It just has like the base of what's going on with it. So it will have what do you guys think it'll have? We'll have anti-A, anti-B, or none of them. Or both of them. It has both of them, anti-A and B. Because it sees both of these as foreign. And then if we have an AB blood type, which my sister is AB positive, so then you'll have no antibodies again. You don't have anti-A or anti-B. Both of those are self to you. So you shouldn't have antibodies like that. So if you are AB, You are what we call a universal recipient. I'm not going to spell everything out. You guys know what this is referring to. So I don't know if you guys can hear that. That's my sister texting me. Basically, AB, you can give them anything. They don't have antibodies against anything. If you are an A, though, you can only give them A. And what else? There's one other thing. You probably think it's AB. It's not. It's not, because they'll react against the B in the AB. So all you can give them is A and O. O doesn't have anything on its surface. Think of it that way. B can be given B and O again. So what can O get? O can only get O. So this one, because it can only get O, but because look at this, A can get this, B can get this, O can get this, and AB can get it also, because it's the universal recipient, it can get any kind of blood type. That makes O the universal donor. Any blood type can receive O. Okay, so I hope that's broken down for you guys to see and understand a little bit. Then we have the RH groups. That RH refers to the rhesus factor. It's just another marker on the red blood cells, whether you have it or you don't have it. If you are positive, RH positive, you'd be like A positive, which I'm A positive. I have the RH factor on my cells. That just means I have that marker on my cells. And if you are RH negative, you just don't have the marker. So if you are a person, a mother, who has a child growing in your belly that was made via a father that was RH positive, and now the baby's RH positive growing in the mother's womb, the mother's body will see the baby's blood as foreign, as an antigen, okay? It's not self, because that RH group, and it interacts with the baby's blood just via the placenta, right? The baby doesn't develop blood until later on in development in the womb, so she doesn't react to it right away. In fact, on her first pregnancy, she might not even know that she is RH negative and the baby's RH positive, and there is a reaction going on there. It's with the second baby that it becomes an issue. That can form what is called hemolytic disease. It's what it sounds like. We already talked about complement-mediated lysis, so we're literally going to be breaking open the cells of the baby that's growing in this mother's belly. So second go around, she has memory now. So once that baby gets to a certain point in development that it starts having its own blood, and the mother could potentially interact with it via the placenta, she will almost instantly start reacting by producing antibodies that fight against the baby's blood, just like you would if it was a disease that you're seeing for the second time. So that would lead to massive destruction of red blood cells for the baby, and can lead often to death of the fetus that way. This still happens today. This is not an uncommon thing. And this is what RhoGAM helps with. RhoGAM helps prevent the mother's body from attacking the baby. Okay, type 3, this deals with immune complexes. So you guys remember how in the previous chapter, let's see if I still have it up. Yes, I do. We talked about agglutination and how all these antibodies can stick onto all of these bacteria and make like basically a little clot of bacteria and antibody and all over, and they're all stuck together like a mass, right? Well, big complexes like that should be cleared by the body pretty normally. However, some of the kind of smaller ones, they can stick around and not be caught by, be cleared out very quickly. So they're floating around, maybe after a recent infection or something. They can get stuck in like tight spots like capillaries. They can get stuck in the endothelium of the actual blood vessels. And then we can have responses via the like neutrophils and everything like that come along and see the cell sitting there and start to react in an inflammatory way, because they're like, oh, hey, here's antibody. Well, that means we react to this. And then inflammation ensues. And so we see this a lot in places like joints and capillaries that are very thin, like in the cheeks, right? So this is dealing with SLE, which is lupus. And you remember the butterfly rash for lupus, that's from this reaction. You can also get it in the joints, and that's what we see with rheumatoid arthritis. Type 4 hypersensitivity reactions, these are delayed. They're cell mediated, and these are related to the T cell response. So up until now, types 1, 2, and 3 had to deal with antibody. Type 4 now, we're dealing with the T cell response, specifically the cytotoxic T cell response, the CD8 positive cytotoxic T cells. These are the ones that go out and typically fight against endogenous pathogens, right? So this can go wrong a little bit whenever we start having reaction via that, but to things that we don't want it to. So that could be reacting to, what's a good example? Poison ivy, right? Oh, it's on here. I just read it. Poison ivy is a very good example, right? You first get poison ivy the very first time. You actually probably didn't know it the very first time because you had to get sensitized to it, just like we do everything else. You had to see it the first time. It's the second time, the memory time, that things go wrong. That's with a lot of these that we're talking about in this chapter. So that's the case with contact dermatitis like poison ivy, and that can include other types of dermatitis as well. And a lot of times if you have allergies that are happening against like latex or something like that, it's not only IgE in the type 1, it's also type 4. They can both be happening at the same time. So these guys, because they're reacting to the same antigen, the body already saw that as foreign. This also includes graft rejection. Anytime that you get a transplant from somebody else and their surface markers don't exactly match yours, basically their MHC complexes will be presenting, well, actually, they don't have to be presenting anything. Our cells, our CD8 T cells, our cytotoxic T cells will interact with their MHC just by itself on like the liver that you just got and just attack it upright, because it's just like outright is what I meant to say. And just attack it because it sees it as foreign, because to them that MHC, the actual MHC molecule, that presenter molecule, that itself looks weird. So it just starts attacking cells like that. So anytime that we are doing trying to match people to their own kidneys or whatever, we're trying to make sure their MHC molecules or HLA molecules, which are just markers for this type of thing, make sure that they match up as much as possible. I used to work in a transplant immunology lab. This is what I used to do for a living. And it was very stressful, and I don't want to go back. But yeah, so there's all different kinds of grafts that you can receive. Anytime that you receive tissue into one area from another area, whether it's from you or something else or someone else, that is called a graft. So you can have an autograft from yourself, an allograft from somebody else that's not related to you, or it could be related, but it's not a twin. Isografts are from twins, so they should be exact matches. Well, roughly exact matches, right? As close as we can get, not being self. And then right, you said allografts already. Like if your sister gives you a kidney or something, or a stranger. And then xenografts, that's grafts that come from other animals. And this sounds weird right up front, but like, you know, pig valves for the heart or something. It's not that weird. That happens all the time, actually. So that, we talked about that. Yes. So whenever you receive a graft, let's say it is a kidney, and your CD8 positive T cells, your cytotoxic T cells, attack that kidney because it isn't a match, because the MHC1 molecules look weird. Basically, that is us attacking the graft. So that's host versus graft. If you get a bone marrow transplant and that bone marrow didn't match enough, then remember, bone marrow is where our lymphocytes actually come from and originate from. So then you can have the donor's bone marrow actually creating cells that can attack your cells. So that is graft versus host. So that's the difference between those. Moving on to the immunodeficiencies or the hyposensitivities. Primary, this is anything that you get from birth, so it's genetically related. The lecture we talked about SCID, severe combined immunodeficiency. It can be often X-linked, so it's usually involved with men because it's associated with the X chromosome, and men only have one X chromosome, so they don't have an alternate X chromosome to fall back on for the proper gene for it. So men tend to be the ones that have SCID. So that's bubble boy. if you guys have heard of that. These people have no immune system at all. They can react to nothing, no memory whatsoever, and they can't even fight off an infection at all. So that's why they live in the bubble, and that's why they don't live very long. It'll be the worst. You can't get out of it ever. So secondary immunodeficiencies, we have the acquired versions. So this is anything where it could be like chemotherapy or radiation, or it could be due to an infection like with HIV. So yeah, so that's basically the rundown of the immunopathology that we talked about for chapter 16. And I hope that this was fun and interesting for all of you. And we're almost through the first half of the second half of the course. So that is essentially what equates to what my traditional students in the fall and spring semester, this would be unit 3, and we're almost to the end of unit 3. So cool.